Here we are in the last video of the Unit 3 series, which covers cellular energetics, and this is video 5, which is focused on cellular respiration, although it's also going to touch on fermentation as well. So before we dig into the details of either of these processes, what is the overall goal of respiration and fermentation? And to put it in context, how does it relate to the picture shown here? Cellular respiration and fermentation are processes that are characteristics of all forms of life. And what they have in common is that they are processes that provide energy for our cells to use from the organic molecules that we eat and drink. So the way this relates to this slide is you can see this woman is using energy. Looks like she maybe just exercised, so she might have been using her muscles a lot, but even just staying still, even just sleeping uses energy. We need to replenish that energy from the organic molecules that we take in because we can't photosynthesize, so we need to get that energy from somewhere. So during this whole video, I want you to be thinking about how is it that we can extract that energy from the organic molecules that we eat and drink and turn it into usable energy for our cells. To understand respiration and fermentation, we need to understand the role of oxygen in these processes. So in the diagram on the left, we see some organisms, those little red circles, that might be located in different areas depending on their relationship with oxygen. So some you see at the top of that growth medium, some are at the bottom, and some are spread out throughout it. So these positions relate to their sort of lifestyle strategies of these organisms and relate to the terms obligate and facultative, as well as aerobic and anaerobic. So see if you can define those four terms right now. So obligate means that it has to, that's sort of the only choice it has. It is required. Facultative is, it's possible, so it can kind of go either way. Aerobic means oxygen, anaerobic means without oxygen. So on the left we see obligate aerobes. These are organisms that can only survive in conditions of oxygen being present. Obligate anaerobes are organisms that can only survive in conditions where there's no oxygen present. And then facultative anaerobes are organisms that can kind of survive in either place. So you see a lot of them clustered towards the top where there's more oxygen, but they can also survive in conditions where there isn't any oxygen. Now, an organism's relationship with oxygen has to do with its energy acquisition strategy. So three major strategies of getting a usable form of energy out of organic molecules is aerobic respiration, anaerobic respiration, and fermentation. Aerobic and anaerobic respiration are both forms of cellular respiration. Cellular respiration is a form of energy acquisition that involves an electron transport chain. The difference here is whether or not oxygen is required. In aerobic respiration, oxygen is required. In anaerobic respiration, oxygen is not required. These are different from the process of fermentation, which doesn't have an electron transport chain at all. So this enables organisms to get a bit of ATP from their organic molecules without having any oxygen and without even needing to involve an electron transport chain. We'll look in more detail at aerobic respiration and fermentation. We're not going to focus on anaerobic respiration. So let's begin our deep dive into the process of aerobic cellular respiration. To start, let's see how much you already know. I want you to see if you can name any of the 5 steps shown here. The 5 major steps of aerobic cellular respiration are glycolysis, link, Krebs cycle, electron transport chain, and chemiosmosis. Glycolysis occurs in the cytoplasm of the cell, and the other reactions occur within the mitochondria. The glycolysis step and the Krebs cycle step both produce a bit of ATP, but we get most of our ATP in chemiosmosis. We will go through each of these processes in detail, starting with glycolysis. So on the left you see a diagram of glycolysis. Let's start by focusing on the reactant and the products, and then answer the question, is glycolysis anabolic or catabolic? So the reactants of glycolysis are glucose, NAD+, as well as ADP and a free phosphate group. What we get out of that are 2 molecules of pyruvate, NADH, and ATP. Let's take a closer look at this process. So we start with glucose, which is a 6-carbon molecule. we end up with 2 molecules of pyruvate, which each have 3 carbons. So when we're going from one 6-carbon molecule to 2 3-carbon molecules, this is essentially a breakdown reaction, which means that it's catabolic. So we're taking a larger molecule and making smaller molecules. Along the way, we are going to produce some NADH, which is an electron carrier, as well as ATP. You might notice that ATP is used in the first part of this reaction to kind of kickstart it, but then we end up getting more ATP out in the end. So the net gain in glycolysis includes some ATP. This ATP is produced as part of an enzymatic reaction. And because it's part of an enzymatic reaction, it's called substrate-level phosphorylation. So that just means that it's phosphorylation, or sticking a phosphate group on ADP to make ATP, that's part of another process. It's part of an enzyme-driven reaction. Substrate-level phosphorylation is in contrast to oxidative phosphorylation, which we'll see later in this presentation. So the next step of cellular respiration is called the link reaction. Take a look at this diagram and figure out what the products are of this reaction and what happens to those products. So in the link reaction, we have pyruvate, which is a 3-carbon molecule, and that splits into carbon dioxide, which is a 1-carbon molecule, and acetyl-CoA, which is a 2-carbon molecule. So once again, we have a catabolic reaction where we're going from a larger molecule to smaller molecules. Carbon dioxide is simply released as a waste, whereas acetyl-CoA is going to be further broken down, so that becomes a reactant later on in cellular respiration. We should also note that NADH was produced here, and that's going to be useful in the ETC as a later step of cellular respiration. The next step of cellular respiration is called the Krebs cycle. This is also sometimes referred to as the citric acid cycle. So what are the reactants and products of the Krebs cycle? One of the reactants of the Krebs cycle is acetyl-CoA. That's a 2-carbon molecule that was produced in the link reaction, and we can see that at the top of this cycle. So it enters the cycle, and then through a series of reactions that you really don't need to know the details of, we end up with 2 molecules of carbon dioxide being chopped off and released as waste. And as we proceed through this cycle, we also produce some things that we're actually going to need in the later steps of cellular respiration, which are electron carriers and ATP. So the electron carriers produced by the Krebs cycle are NADH, as well as FADH2, which means that in order to make those, we need electron acceptors, which is the NAD+ and FAD. We also get a bit of ATP from the Krebs cycle, and so as a reactant, we have ADP and phosphate, and we end up getting a bit of ATP through substrate-level phosphorylation. Before we go on to the next step of cellular respiration, I want to make sure that we have a chance to review glycolysis, link, and Krebs. So, for each of these, what's the overall process that's taking place during these 3 stages? What are the products? And which products are important for the next steps of cellular respiration? So the overall process here is the stepwise breakdown and oxidation of glucose. And we do this in steps in order to better efficiently extract the energy from that glucose molecule as we break it down one step at a time. This produces the electron carriers, NADH and FADH2, and those are going to be used in the electron transport chain. We also produce a little bit of ATP, and carbon dioxide is released as waste. So if you take a look, we start with a 6-carbon molecule. That's broken down in glycolysis into 2 3-carbon molecules of pyruvate. The link reaction further breaks down those molecules, so now we go from pyruvate, a 3-carbon molecule, to acetyl-CoA, which is a 2-carbon molecule. As that happens, we release some carbon dioxide. And then we enter the Krebs cycle. And in the Krebs cycle, acetyl-CoA gets further broken down into 2 molecules of carbon dioxide. Now that we have those electron carriers, we can use them in the ETC. But before we get too far into it, let's make sure you remember what ETC means. Define ETC. ETC is the electron transport chain, and that's the transfer of high-energy electrons from one molecule to another through a series of redox reactions. The electrons lose energy along the way, but an H+, or a hydrogen ion gradient, is produced. And you can see that happening in this diagram in the reactions labeled as 4. So we have our electrons starting from NADH, and FADH2 is also used in cellular respiration. But so the electrons from these electron carriers are transported from one molecule to another through a series of redox reactions, and you can see that hydrogen ion gradient being built up. So where this is happening is really important. So let's look at that top picture up above for reference. So the hydrogen ion gradient is being built up in the intermembrane space. That's the space between the outer membrane of the mitochondria and the sort of folded membrane of the mitochondria. So that's a relatively small space where these hydrogen ions are being pushed into. To fully understand cellular respiration, you need to be able to keep track of these electrons. So where do they end up? What is the terminal electron acceptor in cellular respiration? The terminal electron acceptor in aerobic cellular respiration is oxygen. So keep in mind that oxygen is really electronegative. It's in its diatomic form. It's very hungry for those extra electrons. And so the ETC relies on that sort of high electronegativity of oxygen to essentially pull the electrons along the electron transport chain. And this is why we breathe. The reason that you breathe is to take in air that has oxygen in it so that that oxygen can go to our cells and be used as part of cellular respiration. So understanding that oxygen is that terminal electron acceptor is really important. Following the electron transport chain, the next step of aerobic cellular respiration is chemiosmosis. We've talked about this before in photosynthesis, but let's review. What is chemiosmosis? Chemiosmosis uses the energy stored in a hydrogen ion gradient across a membrane to drive cellular work, in this case ATP synthesis. So on the right side of this diagram, you can see the enzyme ATP synthase, and that enzyme uses the power of the hydrogen ion gradient to phosphorylate ADP into ATP. So let's put this all together. What is oxidative phosphorylation? So oxidative phosphorylation is the combination of ETC and chemiosmosis in cellular respiration. Textbook definition is it's the production of ATP using energy derived from the redox reactions of an electron transport chain. You can compare this to photophosphorylation that we saw in photosynthesis. In that case, it was the combination of ETC and chemiosmosis set up by the light-dependent reactions of photosynthesis. So here we have a very, very similar process, but in this case, it was the energy from the food we eat that provides the energy of the ETC, that provides that hydrogen ion gradient. But in both cases, we have chemiosmosis using the hydrogen ion gradient to produce ATP. So to continue this comparison, let's look at kind of a side-by-side diagram, a really simplified diagram of a mitochondrion and a chloroplast. So see if you can figure out what these symbols represent in the diagram. So the rectangle shown as part of the membrane represents the ETC, and that kind of lollipop shape there is ATP synthase. That's that enzyme that uses the gradient to produce ATP. And the red arrows are showing the flow of those hydrogen ions. So as a review, last time, what is chemiosmosis? So chemiosmosis, as a general definition, is movement of ions across a semipermeable membrane down their electrochemical gradient. So how is this similar and different in cellular respiration and photosynthesis? So in both cases, in both the mitochondrion and in the chloroplast, we have a hydrogen ion gradient created by an electron transport chain. This gradient builds up in a region that has a high surface area to volume ratio. So it's happening in an area that has a lot of membrane, but a very small enclosed space within that membrane. In both of these cases, we have a passive movement of hydrogen ions down their gradient, which powers the production of ATP. It powers the phosphorylation of ADP to create ATP. And in both cases, we're using the enzyme ATP synthase. Now let's look at some differences. So one of the differences is where this takes place. In the mitochondrion, the ETC is on the inner membrane, and the hydrogen ion gradient is built up in that intermembrane space. In the chloroplast, the ETC is on the thylakoid membrane, and the hydrogen ion gradient, or the large concentration of hydrogen ions, is built up inside of the thylakoid. The other difference is where are we getting the initial energy. In the ETC of photosynthesis, the electrons get energy from light. In the ETC of cellular respiration, the electrons get energy from the oxidation of organic molecules. The combined processes of ETC and chemiosmosis are called photophosphorylation in photosynthesis, and they're called oxidative phosphorylation in cellular respiration. Now that we've gone through the process of cellular respiration, I want you to take a look at this diagram, and I certainly don't want you to get overwhelmed by the details. I want you to find one major takeaway from this diagram. So what I want you to see from this diagram is that our bodies are able to break down lots of different molecules, not just glucose, and much of the same machinery is used in the breakdown of other molecules. So just be aware that carbohydrates aren't the only molecules that can be broken down for energy, and many of those processes are exactly the same. All right, the last topic for this unit is fermentation. So if you remember back at the beginning, fermentation is an alternative process to cellular respiration. Under what conditions does this happen? What's the function of fermentation? And why is it that most organisms favor respiration over fermentation when there's the choice? Fermentation is a process that occurs when there's no oxygen available. So fermentation allows for the continuation of glycolysis. If you remember that glycolysis produces a bit of ATP. Now it doesn't produce nearly as much ATP as the later steps of cellular respiration, like chemiosmosis. But remember, in aerobic cellular respiration, chemiosmosis relies on that oxygen being the terminal electron acceptor of the ETC. So if we don't have that, we need to find some other way to keep going, right? If the demands of our cells for more ATP outpaces the amount of oxygen we can supply them, we still have a fitness advantage of getting extra ATP to them. And the way we do that is fermentation. And so fermentation resupplies NAD+ to allow for more ATP to be made through glycolysis. Now, it doesn't make as much ATP as respiration, which is why generally organisms favor cellular respiration over fermentation, but it's better than nothing. A little ATP is better than no ATP because it can keep those cells going for a little bit longer, do more cellular work, and give a chance for that oxygen to be resupplied. So where does fermentation happen? Well, glycolysis is common to both fermentation and respiration. Remember, glycolysis happens in the cytoplasm of the cell. Fermentation, which happens when there's no oxygen present, happens in the cytoplasm of the cell, whereas most of the other steps of cellular respiration occur within the mitochondria. And that's it. That's the end of this video, and that's also the end of this unit. So we have reached the end of Unit 3, which was cellular energetics.